Welcome to the final video in this first week module in the course. And in our last video, we talked about cell types of the immune system. And now we're going to close this module, zooming out a little bit and thinking about the tissues and organs that make up the immune system, the physical locations in the body where all those cell types arise and where they interact in order to mount an effective immune response. So if we think about the tissues of the immune system, we can sort of categorize them in two major categories. You can see here that there are organs known as primary lymphoid organs, and they include the thymus and the bone marrow, which are both labeled in red, and we'll learn more about what these two organs or tissues do in just a moment. But we distinguish the primary lymphoid organs from the so-called secondary lymphoid organs, which include the spleen, structures called lymph nodes, as well as structures called mucosal associated lymphoid tissue, or MALT. We're going to break each of these different organs down as we go through the lecture today, but I want to draw your attention to this cartoon diagram, which not only show these organs, but hopefully you can see that they are connected by a vascular network, a set of tubes, basically, that run throughout the body, through which flows a substance called lymph, and this is where we get the idea that these are lymphoid organs. So the flow of lymph between the lymphoid organs is key for many immunological responses, and so we're really sort of learning about the lymphatic system in addition to the functions of the organs that you see listed here. The primary difference between primary and secondary lymphoid organs is that primary lymphoid organs are the site of immune cell development, whereas secondary lymphoid organs are the site of immune cell activation. So in the left side of this cartoon, you can see here that within the bone marrow, we have hematopoietic stem cells, as we talked about in our last lecture, and that hematopoietic stem cells, they can become myeloid cells or they can become lymphoid cells. And the immediate thing that you may notice is that both myeloid cells and B cells develop within the bone marrow. Notice they're both completely within this red compartment in the graph here. So all of the myeloid cells that we were introduced to, as well as B cells, complete their development in the bone marrow. However, T cells are unique in that they initially arise in the bone marrow, but while still immature, before they are really fully differentiated, they travel to an organ called the thymus. And it is within the thymus that T cells fully differentiate and mature into what we would think of as a mature T cell. And in fact, T cells, the T in T cell actually comes from thymus due to their association with the thymus. B cells, it's sort of convenient to think of B cells being associated with bone marrow, although that is not the original designation of that name. There are some species, particularly of birds, that have an organ called a bursa, where their B cells develop. So that's just a little bit of a factoid, so you can associate where the B and the T come from. But again, in mammals and in humans particularly, B cells develop in the bone marrow, while T cells finish their development in the thymus. But no matter where an immune cell develops, it really encounters antigen or it encounters infection of some kind, typically in a secondary lymphoid organ, or it encounters signals that mediate its full activation. And so you can sort of see that here in blue. Activation includes a variety of processes like turning up the processes of antigen presentation, the induction of proliferation, the cell dividing itself so that there are more copies of it to sort of go more places in the body, fight infection more fully. The processes of phagocytosis and then presenting antigen to T cells to fully activate them. Most of these processes happen in the tissues that we refer to as secondary lymphoid organs. Again, the spleen, the lymph node, and the mucosal associated lymphoid tissue, or MALT. And I mentioned that things are moving around between these organs, and as we're going to see, things move around between these different organ compartments, both through the cardiovascular circulation, meaning through the blood, as well as through the lymphatic circulation, through the lymph. And so let's think about the circulatory system a little bit, sort of as the highway through which these immune cells are moving. So as you may remember from previous coursework, you have a cardiovascular system where blood is being pumped by the heart, and in your cardiovascular system, you sort of have an arterial side moving away from the heart. That blood is oxygenated and nutrient-rich, delivering oxygen and nutrients to tissues. And then you have a venous side where blood has been deoxygenated, and you're moving it back towards the heart to be reoxygenated. And between the arterial circulation and venous circulation, shown here in red for arterial and blue for venous, blood moves through tissues through a progressively smaller caliber network of vessels known as capillaries. And one thing that's different between capillaries and arteries and veins is that capillaries are fenestrated, meaning that they're not completely watertight. And in fact, they sort of lose a little bit of the liquid, the plasma in the blood, as they sort of penetrate deeper and deeper into tissues. Most of that liquid still passes through and rejoins the venous circulation and goes back to the heart. But a good portion of the liquid actually falls out of the cardiovascular system, if you will, and is collected in the tissue. And within the tissue, we refer to it then as interstitial fluid. And as you may know, we need interstitial fluid for a variety of reasons. We sort of need water, fluid all over our body for a variety of homeostatic processes. However, blood is constantly circulating in the body, and if fluid is just constantly leaking out of the capillaries and the capillary beds that are in all the tissues in our body, that would be a problem, right? Because eventually, if that blood would just continue to pool and collect, and we would have edema, we would just swell up. We need to get that fluid back into the circulatory system eventually somehow. So there needs to be another way to drain interstitial fluid out of the tissues and return it to the cardiovascular system. The alternative system that accomplishes that is the lymphatic system, which is shown here in green. So you can notice that in addition to the cardiovascular capillary network within a tissue, there is sort of a mirror image capillary or a vascular network of small lymphatic vessels that are nearby. And these lymphatic vessels themselves are also fenestrated, and fluid actually, instead of coming out of them, actually flows into them within the tissue. So interstitial fluid drains into the small lymphatic vessels in tissues near capillaries and flows through larger lymphatic vessels downstream. And as you may have seen in the last slide, sort of every so often along lymphatic vessels, you have these like swelling structures that you kind of see here. And these are things that we call lymph nodes. And lymph nodes are literally just sort of nodes in the network of the lymphatic system. And as we're going to see, these are actually small organs where a lot of immunological processes happen. Lymph nodes, you sometimes hear them referred to as draining lymph nodes. That is because they are draining the interstitial fluid, and once that interstitial fluid enters the lymphatic circulation, we then refer to it as lymph. So lymph starts its life as blood plasma, it intermediately becomes interstitial fluid, and then once it's drained out of the tissue, we refer to it as lymph after it enters the lymphatic system. And it's draining in a real sense. All the tissue where the interstitial fluid is really, it's collecting the molecules, and if there's an infection, it's collecting byproducts and sometimes even whole pathogens themselves from that tissue. And they're going to move with the interstitial fluid through the lymphatic system, and they will eventually find themselves, those molecules will find themselves in the next lymph node downstream. So we can think of that lymph node as draining that tissue and sort of scanning it, surveilling it for any molecules or pathogens or other signs of infection or injury that might be there. We'll see how that works in more detail. But for now, let's keep thinking just about the flow of fluid through this system. So lymph is going to pass through draining lymph nodes, but it is going to come out the other side. And ultimately, we need that liquid to return to the cardiovascular circulation. And so we have a particularly large caliber lymphatic vessel known as the thoracic duct in the sort of thoracic part of the body, which you can see here. And really, all lymphatic vessels eventually empty back into the thoracic duct. And the thoracic duct itself shunts that liquid into structures called the subclavian veins, back into the venous circulation. And this is, in fact, quite close to the heart. And so that liquid can actually be returned to the heart fairly quickly, where it can be reoxygenated and serve as blood until it sort of completes this loop again. So very important to be thinking about the flow of fluid through the body as a major part of how the immune system functions, and that goes hand in hand with the cardiovascular system. So it's not only that immune cells move through the blood, but they also can move through the lymph, through the lymphatic tissues, and a variety of chemical messengers associated with immunity do so as well. So let's think then, we talked about these capillary beds being in tissues. Let's think about that a little less abstractly. Here, let's imagine that we are looking at the cross-section of a skin, some skin on a human body, and that skin has some sort of wound, some kind of scratch, and that bacteria are coming in through that scratch, and they've entered kind of the subcutaneous, the dermal space. And this is very common when you have some kind of scratch, then an infection might come in along with it. Within the tissue, there are going to be a variety of things happening. One thing that we haven't really covered extensively yet is that most tissues have a population of innate immune cells that are just sort of living there and ready, poised to respond to infection that they detect through pattern recognition receptors. So in this example, these are specialized dendritic cells that live in the skin called Langerhans cells, and we're really just using them as an example for the purposes of this slide. But they can detect pathogen-associated molecular patterns associated with these bacteria that are coming in. That's going to activate them, and it's going to cause them to do several things. One is that they're going to start phagocytosing these bacteria, as well as taking up some of these sort of loose molecules associated with the bacteria through processes like macropinocytosis. That's going to load them essentially with antigens associated with this infection. And that's helpful because dendritic cells are very good at antigen presentation, as we've said. However, there are no T cells really in the skin, and so right now they have the antigen, but they're very far, they're very distal in space from where the T cells are. So how do they get from the site of infection where the antigen is to where the T cells are in order to present the antigen and activate them? Well, activation of a dendritic cell through pattern recognition receptor activation induces them to become activated, and not only to start the process of phagocytosis, but also to start the process of migration. And they will actually enter the lymphatic vessel that you can see here in green and move with the flow of interstitial fluid downstream until eventually the dendritic cells, along with other things that are draining out of the tissue, again, like some of these soluble antigens, soluble components of the bacteria that are just sort of loose and floating around in the interstitial fluid, all of that is going to move to the nearest lymph node, and we would refer to that as the draining lymph node, the nearest lymph node in the network of the lymphatic system. And inside this lymph node is where these dendritic cells are going to be able to present that antigen to T cells in order to activate them, in order to sort of fight this infection eventually and mount an adaptive immune response. So let's look at the lymph node and especially the anatomy of the lymph node. What you can see here is that the lymph node has multiple inputs and outputs, and they both from the lymphatic system as well as through the cardiovascular circulation. We were just looking at dendritic cells and soluble antigens flowing through the lymph, and as you can see here, they are going to enter the lymph node through structures that we call the afferent lymphatics, afferent with an A. And they're going to sort of percolate through these different tissue compartments. They're going to encounter different cells and different signals as they go. But eventually that fluid is going to leave the lymph node because, again, it needs eventually to return to the thoracic duct and to the subclavian veins. And so lymph enters the lymph node through the afferent lymphatics, but it leaves through the efferent lymphatics, efferent with an E. And we'll come back to the efferent lymphatics in just a second. So this is the route by which APCs like dendritic cells and antigen are entering. However, lymph nodes and all secondary lymphoid tissues are very enriched for cells that we refer to as naive lymphocytes, so B cells and T cells. And naive refers to the fact that they have never before seen their antigen. They're naive to it. They've never encountered it. They've never seen it before. And typically, activation of a naive lymphocyte is sort of the first part of an adaptive immune response. They need to see their antigen in order to begin to become activated and respond to it in the various ways that different lymphocytes do. Naive lymphocytes are highly enriched in lymph nodes and other secondary lymphoid tissues, but they do not enter through the lymphatics. Instead, they enter through the circulation, the cardiovascular circulation, through structures known as high endothelial venules, or HEVs, so the blue structures that are shown here. So they're going to enter through the high endothelial venules. They'll take up residence in different compartments within the lymph node, what we'll see on the next slide. But once they encounter antigen within the secondary lymphoid tissue, in this case the lymph node, they will then become activated. And activated or antigen experienced lymphocytes will then leave the lymph node through the efferent lymphatics. So something that's key on this slide is thinking about where and when different types of cells and antigen come into the lymph node versus where and when they leave. Just thinking about the flow of cells, the flow of information, essentially, in the process of an adaptive immune response. Now, though, let's look at more closely at the structural anatomy of the lymph node and think about where some of the key processes of antigen presentation and lymphocyte activation are happening. So if we look at one of these structures within the lymph node, I've sort of zoomed in here. And again, this is a cartoon, so it's a little bit of an abstraction. But if you looked at a real picture under a microscope, it really would not look too different. There's sort of all these different layers within the lymph node. And simplifying a little bit, again, for our introductory class, a few key things that I want to point out are that naive B cells are enriched. They're found in great abundance in structures called primary follicles. And the primary follicle here is sort of this darker purple color that's sort of ringed around the pink. The primary follicle is where we're going to find naive B cells that have freshly developed in the bone marrow. They're going to come into the lymph node and take up residence in the primary follicle. In context, naive T cells, they tend to take up residence in a structure called the paracortex, which is a little bit distant from the primary follicle. So there are distinct compartments. The naive B cells and the naive T cells are sort of segregated. They don't sort of intermingle very much in the naive state within the lymph node. So important to know these anatomical compartments. And if we think about then how do naive B cells and naive T cells see their antigens while they're residing in these compartments, it would look something like this. As lymph is coming in to the lymph node, it's going to sort of filter through these different compartments. It's just moving along with the flow of the interstitial fluid. And soluble antigens that are just kind of floating around, not being presented by any particular cell, soluble antigens are captured by naive B cells. And a point that I haven't made yet is that B cells actually do not require antigen presentation, or I should say, they don't require antigen to be presented to themselves. They are capable of directly interacting with antigen, which is why they can be activated by soluble antigens that are just floating around in their environment. And that is indeed what happens. Naive B cells capture soluble antigen as it comes in the lymph. And when a naive B cell finds an antigen that it matches, that its B cell receptor is specific to, that is going to induce it to become activated. And when it becomes activated, it moves into this pink structure that you see here, which is called a germinal center. It would be better to say, actually, that this pink structure is formed following the activation of a B cell, because it's going to sort of move deeper into the follicle and begin to rapidly proliferate, to divide, to make many, many daughter copies of itself. And that focus of rapidly dividing B cells actually is something that we refer to as a germinal center. It's visually distinct, actually, under the microscope. In contrast, remember that T cells cannot detect soluble antigens. They require antigen presentation in order to become activated. Therefore, we need antigen-presenting cells like dendritic cells. And remember, the dendritic cells are also coming into the lymph node through the lymph, and usually they're carrying antigen with them that they've picked up in some other location in the body. Or maybe they've just picked it up from the soluble antigen that's entered. But in whatever case, we need that intermediate cell type, like a dendritic cell, to present the antigen via an MHC interaction to the naive T cell. And when the naive T cell becomes activated, it's also going to begin to proliferate. However, there's no analogous structure. T cell proliferation is also still happening in the paracortex, and there's not something equivalent to a germinal center that T cells form. They're just a little bit more loosely organized compared to B cells. So in both cases, both naive B cells and T cells, they see their antigen, they become activated, and they begin to rapidly proliferate. For B cells, we refer to that anatomical site of proliferation as a germinal center, whereas for T cells, it's a little bit less anatomically organized, but the same physiological processes, more or less, are beginning to happen. So these are the very basics of what happens when naive lymphocytes encounter antigen in the lymph node. And a key point that I want to make here is that lymph nodes are really how we detect infections in most of the tissues of the body. We have antigen-presenting cells that have established residence in different tissues in our body, like the skin, like the liver, the gut, different places where we might encounter an infection. That interstitial fluid is going to drain through the lymphatics and eventually go into a lymph node where we're going to be able to detect an infection through that information flowing through the lymph. However, we can also have infections in our blood, and so we actually detect blood-borne infections through a different organ, not lymph nodes, but an organ called the spleen. The spleen, in fact, is a specialized secondary lymphoid organs for blood-borne antigen. And if we look at a cross-section of the spleen, again somewhat simplified, we can say that there are sort of two major designations or sites within a spleen that we refer to as the white pulp and the red pulp. And just like when we talked about the different types of blood cells, we learned about red blood cells being mostly associated with blood and white blood cells being more associated with immune cells. There's a similar kind of concept here. More like blood-related things are happening in the red pulp. And in fact, the red pulp is rich in erythrocytes, and it does contain a population of macrophages which are sort of eating and recycling old, damaged erythrocytes. For the most part, the red pulp is not really an immunological tissue per se. Most of the processes of antigen presentation and naive lymphocyte activation are occurring in the white pulp, which is much more enriched for white blood cells, leukocytes. And you can see that it is sort of surrounding the arteriole that's coming into the spleen. And so instead of lymphatics bringing fluid and antigen and cells into the lymph node, in the spleen, everything is entering through the arterial circulation, through the splenic artery. So if we think about a sort of cross-section of the white pulp, what are some of the sub-anatomical structures in the white pulp, and how are they analogous to the lymph node? Well, this cartoon is making things quite simple again, but this is really just to understand conceptually the different locations and what's happening within them. Remember that I said everything in the spleen is entering through an arteriole, branching off of the splenic artery. So both leukocytes and soluble antigen enter through the splenic arterioles. And within the white pulp of the spleen, there are a few locations. There is a region called the periarteriolar lymphoid sheath. Periarteriolar meaning around the artery, around the arteriole. And this structure, which is also known as PALS, is rich in naive T cells. So it's roughly analogous to the paracortex in the lymph node. Within the white pulp of the spleen, we also have follicles, primary follicles, just like we saw in lymph nodes, and they are also going to be the main location where naive B cells reside. And then finally, there's an additional zone called the marginal zone. And this is where a large population of antigen-presenting cells, again, like dendritic cells and macrophages, are residing. And they are poised there to encounter antigens that are coming in through the blood, pathogens that are coming in through the blood, to phagocytose them, process them, and then present them to naive T cells. And in fact, you can sort of see that process happening here. Naive T cells come out of the arteriole, take up residence in the pals. Naive B cells taking up residence in the follicles. APCs taking up residence in the marginal zone. And then here, as antigen is coming in through the arteriole, that soluble antigen can directly activate the naive B cells in the follicle, and they will form germinal centers within the follicle, just like we saw in the lymph node. And that soluble antigen can also be taken up by antigen-presenting cells within the marginal zone, which can then migrate close to the PALS region, to the lymphoid sheath where the naive T cells are, and present that antigen to the naive T cells, where they themselves then, again, will become activated and undergo a process that we call clonal expansion. Rapid proliferation of a single naive lymphocyte into many identical daughter cells. This too is going to be a major source of effector T cells that are now activated and specific for the antigen that activated them, that can then leave the spleen and go back into the body looking for that infection and killing any cells, or modulating the immune response in proximity to wherever that infection is happening. Okay, so that's the spleen, but there was one final secondary lymphoid organ that we wanted to cover, and that was the mucosal-associated lymphoid tissue, or MALT. MALT is a little bit hard to pin down because it's so loosely organized. There's not like a picture I can really show you that really has all these discrete structures. It's a little just a bit more loosely organized, as I've said a couple of times, meaning that it's places that have naive lymphocytes, it has APCs, but they're not necessarily so segregated and specifically located in little compartments. They're kind of associated with a variety of tissues in the body that secrete mucus or mucosal surfaces. And this is key because mucosal surfaces tend to be what we might think of as portals of pathogen entry in the body. So our respiratory system, digestive system, our urinary tract, the tonsils and adenoids, these are places where things are entering our body: food, air, bodily fluids from other people, which can carry pathogens. And because these are locations where pathogens are entering our body almost constantly, we sort of have MALT, this mucosal-associated lymphoid tissue, as a sort of first line of defense where we can sort of rapidly, hopefully, respond to any antigens that come in through these mucosal surfaces and mount really quick adaptive immune response, as quick as an adaptive immune response can be. So this is an example of different locations that are mucosal in nature and where we find mucosal-associated lymphoid tissue. Let's just think about one particular type of MALT, just for explanation's sake, for the sake of an example, that we find in the digestive system. The digestive system has a specialized form of MALT that we refer to as a Peyer's patch. And so in a Peyer's patch, it looks something like this. Notice that you don't see follicles, and you don't see like a particular area where a naive T cell is going to be versus an APC. But what you do find embedded within the epithelial surface of the gastrointestinal tract, you have all these epithelial cells which sort of make up the lining of the gut. Embedded within them are specialized cells called M cells. And the purpose of M cells is they sort of survey the lumen of the intestine, the open area inside the intestines, for potential pathogens and antigens. And when they encounter them, they actually take those antigens up and sort of shuttle them through their own cytoplasm in vesicular structures and kind of spit them out the other side into the space of the Peyer's patch. Now, within the Peyer's patch, it is vascularized, and there are high endothelial venules. And through these high endothelial venules, naive lymphocytes, including T cells, can enter. And these naive T cells, while they're hanging around in the Peyer's patch, if antigen is coming in through an M cell, it can be taken up by antigen-presenting cells that, again, are hanging out in the Peyer's patch. And when that occurs, that antigen-presenting cell can activate that naive T cell, and it will then clonally expand and sort of mount its effector functions, again, just like it would if it had been activated in a lymph node or in the spleen. And similar processes can happen with naive B cells within MALT tissues as well. Soluble antigen can come in, they become activated, and they rapidly expand. So Peyer's patches are just one example of MALT, but there are many examples of these loosely associated lymphoid tissues, again, at portals of pathogen entry within our body that tend to secrete mucus. Okay, let's summarize our tour of the different tissues of the immune system. As we saw, there is a very broad network of tissues that orchestrate immune responses, and we saw that we have two different designations of those tissues. We have primary lymphoid tissues, which are bone marrow and thymus, and these are primary sites of immune cell development. And we have secondary lymphoid tissues, the spleen, lymph nodes, and MALT, and these are sites of immune cell activation. Which is what this box is summarizing here. I'll add to that immune cell activation, particularly of naive lymphocytes, is going to require antigen presentation, which again is happening somewhat uniquely to naive lymphocytes within the secondary lymphoid tissues. What we saw is that these different tissue compartments are connected by something called the lymphatic system, which facilitates the movement of antigen and APCs around the body. And antigens and APCs in the interstitial fluid within the different body compartments drain into lymphatic vessels, and then the lymph flowing through those vessels passes through lymph nodes containing naive lymphocytes, where any antigens that are present can be presented to those naive lymphocytes to activate them. When that happens, that is going to induce the processes of activation and clonal expansion. And following their activation, lymphocytes can then leave the SLOs to travel to the body to exert their various effector functions. So that finishes Module 1, and this is the most introductory material in the course. In our next video, we'll move on to Module 2, where we will look with a great bit more detail at the innate immune system. See you there.